Okay, so thanks for coming. I really like these sort of conferences because you sort of can skip to the chase a little bit. I don't need to convince any of you how amazing seagrasses are. I don't have to talk about ecosystem services and all the brilliant things that we benefit from having seagrasses. And unfortunately, I don't have to tell you about the fact that, seagrasses are increasingly decreasing at a rate that's faster than any natural recovery. So restoration is really becoming something that we have to think about as well as protection, but we really do have to start thinking about restoration in just about every part of the globe. Now, there's been a number of brilliant reviews, and some of them have been mentioned, and I think there's three main things that we can get from some of those reviews. Some have been touched on already. The first one is that successes through restoration have come through groups getting together and working over a number of years to build that sort of information that then they have the idea of actually what's going on. I mean, people working for tens of years on some of these things in some cases, and they're the ones that are getting the success. So I don't think there's any quick route to this. The other is to actually have networks of people working together and supporting each other and sharing information, just like we're doing today. And another bit of information that's already been mentioned lots by people already in this session is that scaling up and getting to that stage where you're getting those self-sustaining positive feedbacks to allow the meadow to actually sustain long term, and there are real goals. To do this where I am, we started working with plugs and transplanting plugs, and then decided that if we were going to scale up in the sort of environments that I work in, which I'll get onto in a minute, really the most attractive method was going to be looking at seeds. So we started working with seeds basically because there's less donor bed damage, you get more genetic diversity hopefully, and the time and cost of actually collecting seeds and putting seeds out was hopefully a lot less than some of the methods that we've seen. But still not a really good idea as to whether it's going to be more successful. And using seeds, we can take a lot of the sort of like the advice and roadmaps that are already out there from a lot of terrestrial systems for restoration. So in terrestrial systems, there's ideas on strategies, things like advancing restoration, seascape genetics, having protocols for the correct collection, large-scale native seed farming. There's a whole suite of different strategies that need to be in place before you actually go into seagrass restoration using seeds. And amongst these, there's a number of different hurdles. So there's the genetic issues, problems with seed quality, improper placement in the sediment, a whole heap of different hurdles and a whole heap of different requirements for actual seeds, for actual looking at seed germination, seedling emergence, and actually establishing these seedlings in the field. So what we're doing, very slowly, because we're quite a small team, is to actually try and build up some of this information for the local species of seagrass that we have. And this is in Gladstone, and you might have heard Gladstone mentioned in a few other talks in the last few days. So it's in Central Queensland, and it's one of the it's the 5th largest coal port in the world, but it's absolutely nothing compared to Singapore. It's way smaller, and it's also, importantly, it's within the Great Barrier Reef World Heritage Area. I like this picture of Gladstone because I think it sort of captures it perfectly. So here you've got a wharf with some aluminium bars from the nearby smelter. And this is actually sheltering a muddy bay where you've actually got seagrass that's started to colonise. And somewhere, you can spot it down there, there's actually a dugong feeding. And probably that seagrass might not even be there if it wasn't for that wharf. So this is just a quick plug for our workshop on Friday, looking at these sort of engineering approaches to creating seagrass. So the seagrass in Gladstone Harbour is monitored by Chop Water by JCU for the Gladstone Ports Corporation. And because of this, we've got some really good, as you might have seen from Rob's talk, some really good sort of long-term data about what's actually happening in Gladstone. So Gladstone's the middle line in here, and whilst there's been some ups and downs, generally there's been quite a sort of a large-scale loss in seagrasses over the decades. And these are the culprits, it's the usual suspects. This is just from a sort of an outreach thing that we do, but these are the zombies that are causing all the seagrass loss. A bit of fun, but they are real pressures. And the two big ones are really land reclamation. So this is from some capital dredging. This is where they put the spoil from the capital dredging. And they're about to, and this was a seagrass meadow, and they're about to extend out over here for the next stage of capital dredging that's happening. And the other big one is floods, so really large-scale flood events that are becoming more frequent, putting all of this muddy plumes out into the harbour, and this obviously reduces light but also settles on the seagrass and buries it. We've got 5 different species of seagrass, and the one that I'm going to talk about today, so this is my plants versus zombies, is this one here, so Zostera muelleri. And we're quite lucky because there's already been quite a lot of work on Zostera muelleri. So we already know that the germination rates increase with a decrease in salinity. We already know that this actually varies with temperature a little bit, which might be related to summer and winter germinations. We know that darkness can promote hypocotyl hair elongation, and incision, even though it might be impractical, actually increases germination, and also that germination was faster under anaerobic conditions. But we're really a long way from actually getting to this point where we almost have a protocol and the back of the seed packet type of information to be able to just go out there and put our seagrasses out. And also from when you do go through the literature, you find that this happened in this place, but then if you tried it in this place, it didn't work. So there's a lot of sort of like adaptation variability actually happening just in this one species. We're lucky in the fact that we teamed up with sort of citizen science groups and also the local indigenous sea rangers to go out and actually collect flowers and collect the seeds from those flowers on quite a large scale. So we're getting the number of seeds that we need to do some of the experiments I'll show you. Here you can actually see the seeds. They tend not to be like really long, they tend to be just like little brown sprigs sticking up right close to the mud. But once the people get their eyes into it, they can really go and collect quite a lot. And we can get up to 500 flowers in a square metre and about 12 seeds per flower. So you can really get the numbers, but it's very, very patchy, and we have to sort of continuously monitor meadows to see when the seeds are ready to go and collect, or the flowers. We then put the flowers into our mesocosms and let the seeds just drop out from the actual flowers that we've collected. So we're looking at using these seeds for a number of different questions. The first one that we looked at was what's the best conditions for breaking dormancy, and are they the same across sites? So whilst there's lots of studies out there, we wanted to just check that what's out there actually applied to our site, and also whether there was any within site variation in this. So we took seeds from 2 different locations, Pelican Banks up here and Lily's Beach, and we sterilised them in a sort of a fairly standard way. And we looked at 2 salinities, salinity of 5 and slightly higher at 10. And we exposed these for different durations to these different salinities in either a light or a dark situation. We looked, even though there's lots of different ways of classifying germination, we actually took radical protrusion as being our sort of like stop-off point to say this is what's germinated. And we calculated germination rates using Osborne and Brealey's method. So what we found, we were expecting that there wouldn't really be a difference between sites, but unfortunately there was a significant interaction going on between one of the sites, which we still can't explain, and we're going to actually try and repeat the study to actually see whether it's something that was just an artefact of the study or not. But the main point was that the salinity of 5 was slightly too low, and when we actually raised it to a salinity of 10, we got much better germination rates. The best overall was actually to be in the dark for 24 hours with a pulse of 10 PSU. This also got us thinking about how naturally seeds actually get dispersed, and we've been doing a study looking at seed dispersal, and we started to think about some of the studies that are out there, like Samantha Toll's study and everything, about how seeds are actually moved in different ways. So you've got seeds that actually go along on the surface of the water column, exposed to high light and often in quite sort of like saline conditions, versus ones that are maybe moved in the bedload and bedload transport, things that go in the actual stomachs of dugongs and turtles, and whether any of these actually affected the germination rates of the seeds, and could we learn from nature in terms of how to increase our germination rates. So with this one, we actually, a similar sort of study, but just from one site, exposed to our 10, again, because that worked well, but also to a much higher salinity, and then actually looked at exposing them to slightly different pHs, which were representative of what they would be exposed to in the stomach of one of these mega herbivores. And everything else was pretty much the same. And what we found was that it was significantly higher germination rates when at the lower pH in the dark at 10, which was actually similar to what you'd find in the stomach of a dugong. This one was also high, so these are sort of conditions that they might have if they're in the space floating on the sea surface, but that wasn't actually significantly different from these others because there was a lot of variability. The other question that we had was whether the sterilisation techniques that we were using actually influenced the germination rate. So were we exposing, but you have to in where we live, you have to sterilise them if you're going to keep them from any amount of time, because all the fungi and bacteria grow on them, they get pretty gross. So even when they're in the fridge. So what we looked at was whether this technique actually influenced the germination rates, and looked at the sort of different maturities of seeds. So you go from green to black, basically, in terms of maturation, and whether, sort of treating these sort of greener ones with these sort of bleaches and things like that was going to cause problems. So again, we took just seeds from one site this time and had the 3 different maturations, the different colours of seeds, and exposed them to different levels of sodium hypochlorite solution, and put them again at 10 PSU in the dark for 24 hours, which we found was the best way to germinate them. And what we found that there was no significant effect on germination of the hypochlorite solution, sodium hypochlorite solution, but there was a significant difference between the colours of seeds, which you sort of expect. So it's useful. But the thing with this one is that what we now need to know is that if you've actually treated those green seeds, once they turn black, because they do eventually, looking then at comparing those with the black ones that we get fresh from the plant. And I might just quickly rush through this, but we also then looked at the seedling early survival, just because that's obviously important. This is where a lot of our work's concentrated at the moment. I did know that I'd probably run out of time for this. So we just basically buried them in different depths of sediment, and interestingly found that 5 mils sediment, irrespective of what type of sediment mix you actually put, so sand, mud or mixed sediment, actually had the best seedling emergent rates. What we found with the 0 burial is that as soon as that first leaf came out, they basically just floated off and went through the system, so we actually lost them. So having that slight burial is really, really important to early seedling survival, which is going to be important when we put the seeds out there. So there's a few conclusions, though. They're all very site-specific, and I think it goes back to the talk this morning by Johan, which is like that if we all do these sort of small-scale studies, eventually we'll have enough to do a meta-analysis to really see what the sort of key protocols could be for actually filling in this sort of like back of the seed packet sort of information. And keep building knowledge, and sharing knowledge is my message. Okay, thank you very much. Okay, I'll take any questions very quickly because I think I'm over time. Any questions? Good, okay.